All right, welcome to Chapter 5, The Working Cell. So, this is a chapter all about energy in biological systems. So, let's not tarry any longer. The first thing we're going to talk about is bioenergetics. What is bioenergetics, you ask? That is, simply enough, how energy flows through living systems. So, how energy is used, how energy comes into the system, how energy exits the system. And to talk about energy, we need to define energy. And to do that, we need to separate energy into categories. And there are certainly lots of categories of energy, but we'll just go through 4 types. There's kinetic energy, with my awesome campy animation there. Bam! And that is energy from movement. So, kinetic energy, well, let's see. I can generate some kinetic energy here. What? Moving stuff. It's moving. That's kinetic energy. Then there's thermal energy. Thermal, anyone want to take a guess at what thermal energy is? Huh? That's right. Energy from heat. You're going to generate some thermal energy. There it is. My hands are warming up, so the friction in my hands is generating thermal energy. Next, we have something called potential energy. This one is sort of a little more complicated, but not too terrible. So potential energy is defined as energy possessed by matter because of location or structure. Think about it as energy that's sort of stored. It's not energy being generated. What it is the potential for energy should its situation change. So in general, things that are unstable possess more potential energy. They have a large amount of potential energy if they are unstable. This could include chemical compounds with relatively unstable bonds. ATP, adenosine triphosphate, the energy currency for the cell, consists of an adenine linked to a ribose sugar, which is then linked to 3 negatively charged phosphate groups. Now, every single one of them are negatively charged. And if you put 2 negative ends of a magnet near each other, what do? They repel. So all 3 of these phosphates are trying to repel from one another. And they are linked together by something called a phosphate bond, which, believe it or not, is even weaker than a hydrogen bond. And so that makes ATP highly unstable. So that if you were to break its bond, it would release a lot of energy. By that same token here, I can talk about, say, potential gravitational energy. So this bottle here is precariously raised in the air. And so it's also unstable by the way I'm holding it, and it has gravitational potential energy. That means if its situation changes, it will produce gravitational energy. Now let's take a look at that. There it was. I let go of it, and gravity took over, and it produced gravitational energy by falling. So, next up, chemical energy. Chemical energy is potential energy available for a chemical reaction. So, the energy produced during chemical reactions is chemical energy. So, for instance, when you break a phosphate bond in ATP and you hydrolyze that bond and break off a phosphate, you release a bunch of chemical energy. And in our class, we're referring to biological reactions specifically. All right, so now it's time to talk about thermodynamics. So we've defined a few different forms of energy. Now it's time to talk about how energy works in the universe. Thermodynamics is the study of energy transformations in matter. And there are 2 basic laws of thermodynamics, things that have incontrovertible evidence to support them. The first law of thermodynamics is the conservation of energy, and it states that energy can be transferred and energy can be transformed, but it cannot be created or destroyed. What this means is all the energy that is in the universe now is all the energy that was ever in this universe, and all the energy that will be in this universe till it ends. So, what happens to that energy then? Like if you break the bond in an ATP and release the energy, where does it go? Well, this refers right back to energy can be transferred and energy can be transformed. See, a transfer of energy is relatively easy. When I am moving my hand, I am generating kinetic energy. And if I stop my hand, the kinetic energy is transferred to this bottle. So there we go. That's an energy transfer. And then energy can also be transformed. So if I put my hands together and start rubbing them, I've got kinetic energy and frictional energy, and both of those are going to be converted into heat energy, right? Thermal energy. So that's an energy transformation. Another one, I can use kinetic energy and collide with something. And that kinetic energy produces sound energy. It produces heat and sound. So that's another energy transformation. So we can transfer energy and we can transform energy, but it cannot be created or destroyed. That is the first law of thermodynamics. I love that law because it means my grandfather, who passed away in 1996, his energy may have left his body, but it is with the universe always. So that's kind of comforting to me. Who says science has to be cold? All right. For the second law of thermodynamics, we have to first understand a concept called entropy. Entropy is the tendency for matter in a system to move from a state of order to disorder. What that means is things break down over time. Things decay. Our body is highly ordered, and then when we die, it falls apart. In fact, in order to maintain order, you must constantly be rebuilding something or fixing it. So in the universe, everything has a natural tendency for moving in a system from a state of order to disorder. That is entropy. How does this factor into the second law? Well, the second law states that every energy transfer or transformation increases the entropy of the universe. So, the second law also states explicitly that energy cannot be recycled infinitely. Think of this as a wheel. If you take your hand and start spinning a wheel and then you let it go, the wheel over time slows down and then comes to a stop. That is because some of its energy is lost to transfers and transformations. It cannot consistently recycle its kinetic energy as it moves. It cannot infinitely recycle it. So instead, it slows down and stops. So I always just found this funny, so I'm going to play it because I enjoy it. Clip from The Simpsons referring to the second law of thermodynamics.
Lisa's becoming very obsessive. This morning, I caught her trying to dissect her own raincoat.
I know. And this perpetual motion machine she made today is a joke. It just keeps going faster and faster. Lisa, get in here. In this house, we obey the laws of thermodynamics.
Sorry, I had to talk along with it. I love The Simpsons too much. But I always just like that because they included a shout out to the laws of thermodynamics with Lisa Simpson's perpetual motion machine. which is something a lot of people have always wanted to build. The idea of a machine that can recycle its energy infinitely, so that if you start it accelerating and it retains all the energy that you put into acceleration, then it will just continue accelerating infinitely. So that's how that violates the second law. So now let's kick it back to biology and talk about the kind of work that cells perform. Cells do transport work when they move molecules across the membrane. And it requires energy, it requires work energy. So, yeah, stupid out of order animations. Anyway, work requires energy. And transport work, in particular, requires energy when it is violating the natural rules of diffusion. Specifically, when a transport protein is moving things across the membrane from an area of low concentration to an area of high concentration, called active transport. So active transport requires energy in the form of work. Mechanical work is also done by cells. Mechanical work is relatively easy to think about. The moving of cilia or flagella on a single cell is something mechanical. They're controlled by a simple protein motor. In the case of cilia, the cilia move back and forth, kind of like oars on a ship, and then a flagella tends to rotate, kind of like a propeller. That is mechanical work. And then in something multicelled that has muscles, the muscles themselves are a mechanical machine. So they perform mechanical work, and it requires ATP to do so. Next up is chemical work. Chemical work is pretty much everything else a cell does that requires energy, in that cells perform large numbers of chemical reactions. And chemical work is most often seen with reactions that produce something, synthesis reactions. For instance, when a cell takes individual amino acids and assembles them into a protein. So that takes work. When a cell digests something, or when a cell does large numbers of things, it requires work. So chemical work. Oh no, no animations on this slide. I can fix that. Haha, and you're all stuck around for the ride. Aha! Instant. I'm just so awesome. So chemical work uses chemical energy. And this is energy produced in chemical reactions. And when you use chemical energy, 66% of the energy produced in a chemical reaction is released as heat. So you lose 66% of that chemical energy, leaving just 34% left over for chemical work. So that heat that you lose when you undergo a chemical reaction is why you have body heat. So kind of useful actually, because generating your own body heat allows you to be more active. So how do we measure work? So there's an easy way to measure the amount of energy contained in a chemical, and that is using a unit of measurement called the calorie. And the calorie here is written with a lowercase c. That will be important later on. So a calorie is a measurement of the amount of work done, or the amount of potential energy, potential chemical energy contained within a structure. So you measure the energy used by something using the calorie. In fact, in the food industry, calories are measured by how long it takes something to burn and how hot something burns. So they actually torch food in the food industry in order to determine that calorie number. So calorie is specifically defined as the amount of energy required to raise 1 gram of water by 1 degree Celsius. And kilocalories is the unit of measurement you are familiar with, because the kilocalorie is 1,000 calories, and this is the calorie with a capital C that is printed on the nutrition labels of all your food. So your food is measured in kilocalories. Multiply it by 1,000 if you want to know how many calories that is. But, So, I told you that the case was important. Kilocalorie is represented by the word calorie with a capital C, whereas calorie with a lowercase c is just the basic unit of energy, which is the amount of energy required to raise 1 gram of water by 1 degree Celsius. So, there you go. Moving requires energy. And so that energy can be thought of as using calories. A 7-minute mile uses 979 calories worth of energy. Food provides you energy. For instance, a cheeseburger gives you 295 calories, which means that, of course, if I run a 7-minute mile, I can then eat 3 1/3 cheeseburgers and feel perfectly neutral about it, which is what I do when I possibly can. And then the rest of the time, I like to rationalize eating far more calories than I expend. Something along the lines of, my brain needs the energy, or, I just want to enjoy life, or other crap like that nobody truly believes, but it helps them ignore the fact that they're filling themselves up with extra calories, and it will make them nice and fat. So, meh. Powering work in the cell. ATP is hydrolyzed, which means the bond in between the phosphate and another phosphate is broken. Hydrolyzing a bond in ATP produces energy. So, ATP, for those of you who forgot, is adenosine triphosphate. And it has lots of potential energy. Now, I already mentioned why, because of those 3 negatively charged phosphate bonds trying to repel one another, held together by the relatively weak phosphate bond. So 3 negatively charged phosphate groups repelling another, held together by weak phosphate bonds. That's a very unstable chemical, and so it has lots of potential energy. When it's hydrolyzed, it produces energy. And when it's hydrolyzed, you release a single phosphate. So ATP, adenosine triphosphate, loses one of its phosphates. So I wonder what it's abbreviated as when it only has 2 phosphates. What could be a Latin prefix for 2? Di, you say? You're right! When ATP is hydrolyzed and a phosphate is released, you produce energy and you convert ATP into ADP, which is adenosine diphosphate, 2 phosphates, plus a spare phosphate. So, bam. So let's look at breaking ATP. When you hydrolyze ATP, you break one of those phosphate bonds holding a phosphate group onto ATP. So I just put H2O in there, but this is sort of written kind of crappy because that H2O is not one of the reactants. So you're not going to get one of those substitution reactions where it incorporates the hydrogen and the oxygen to make new molecules. Instead, I put plus H2O because hydrolysis is hydro using water and lysis to break or burst. So you are using water to break the phosphate bond holding one of the phosphates on it. You convert it to ADP plus P, which is an adenosine phosphate, an adenosine diphosphate, plus a spare phosphate. So breaking that bond releases energy. All right. Next up is what we do with our adenosine diphosphate and our spare phosphate. Because a cell requires vast resources in order to be alive. And in order to conserve those vast resources and not just have to be a continuous eating machine to get enough nutrients to continuously crank out ATP, we have to save our ADP plus P. So our adenosine diphosphate and our phosphate group are hung onto by the cell. And when you add that phosphate back to adenosine diphosphate, you recycle it back into ATP. So there you go. What happens to that phosphate before recycling? Because you don't just hydrolyze ATP, immediately get that phosphate loose, and then immediately rebind it. Instead, that phosphate goes to something called the phosphorylated intermediate. The phosphorylated intermediate is, by and large, the structure that is doing the work. So for an active transport protein to function, a phosphate has to bind to it in order to induce the correct shape change to allow the transport protein, the active transport protein, to move something from an area of low concentration to an area of high concentration, thus violating the rules of diffusion. So let's look at that phosphate transfer a little more closely. The molecule doing the work hangs on to the phosphate that was released when we hydrolyzed ATP. And the binding of that phosphate is what allows the work to occur. After the work is finished, then the structure that hung onto the phosphate, the phosphorylated intermediate, releases that phosphate back into the cytoplasm. And that phosphate is recycled by recombining it with adenosine diphosphate in order to reproduce ATP, adenosine triphosphate. So, bam. What would happen if there was no recycling? Well, a human muscle cell uses all of its ATP in approximately 60 seconds. Without recycling, I would have to produce my human body weight in ATP every day in order to stay alive. That's a ridiculous amount of ATP. I did the calculations for you by determining the atomic mass of ATP and then dividing it by my body weight in order to determine the number of ATP molecules I'd need in a day, and that is 1.08 times 10 to the 26th molecules, which is a ridiculously large number. I can try and put it in terms that might boggle your brain a little less, or maybe even boggle your brain more. 1.08 times 10 to the 26th is 108 trillion molecules. So it's 108 trillion of a trillion molecules. It's a ridiculously large number. I would have to be a constant eating machine. And even then, I probably wouldn't satisfy the number of ATP needed in my body, and I would just die. So recycling allows us to live. So you recycle about 10 million molecules of ATP per second per cell. So every cell in your body is breaking ATP into adenosine diphosphate and a spare phosphate. The phosphate is bonding to the phosphorylated intermediate and then released in order to regenerate ATP from adenosine diphosphate and that spare phosphate. And you do that 10 million times per second per cell. That's some pretty epic recycling abilities. Okay, so next up is enzymes, but this seems like a good point to stop this particular part of the lecture. So I will catch up with you in the next video.